All right, so welcome to the research proposal defense of Nathan Lazar. My name is Kemal Sönmez. I'm an associate professor in the department. Nathan came to us with a bachelor's in mathematics from Ohio University and a master's of science in mathematics from Portland State. And I remember first meeting with him. He had run up his bike on the hill, and we kind of didn't meet at the right time, the first day, I guess, but then we connected. And so he came as a master's student first, and then the next year he secured an NLM fellowship. Mathematics is really one of the very, I think, great preparations you can have for this for the kind of thing that Nathan does, which is sort of really modeling at a deep level. So today he's gonna talk to us sort of about what kind of research he's proposing. So he has looked at several things in the past, and now he's sort of working with his committee. He has put together a research proposal. So without further ado, I'll just turn it over to Nathan.
Thanks, Kamal, and thank you all for coming. And thanks to the people that are watching out there, including my father, who's been waiting since 11:30 Eastern Time. Okay, so my proposal here is about these Bayesian tensor factorization method and applied to drug response in cancer cell lines. So I'll just jump right in because there's a lot to get into. This is a 4-dimensional hypercube, just to give you something to look at while I do my outline. So I'll talk about the motivation behind the method, why I'm addressing this problem, some background on this tensor stuff, current approaches towards the cell line drug prediction problem, my 3 aims, and some preliminary analysis of the data that I've done that proves to me or shows to me that this is likely to be fruitful in the timeline for the rest of the project. So just as an overview, this is kind of in the broad category of trying to move towards precision medicine. So we know that basically all patients don't react to drugs in the same way, and certain drugs will be less effective in certain populations of cancer patients. So what we really need to move towards is this idea of precision medicine, where we do some type of categorization of the patient, looking at their genomics or other markers in order to decide on what drug treatment will be most efficacious in that particular patient. So one way to do that, we can't really test a bunch of different drugs on one patient, right? That wouldn't work well. So if we can get some type of smaller system where we can test these drugs in the dish and see which drugs might work for a particular patient, and then move that towards the clinic, that's a lot more of a tractable problem. So one way to do that is to extract cells from the patient and then grow them in a dish. And this is a cancer cell line. These are HeLa cells, which was the first cancer cell line to be developed back in the 50s. And then test drugs on these and see which drugs are likely to work for that patient. If we can do this in a high throughput way, then we can understand more about the relationship between the genetics and the response to drug therapy. So this is one of the smaller 96 well plate from a smaller one of these such experiments, where you can put either the same cell line in each of these little wells or different cell lines in each well, treat them with different drugs at different doses, and then after they're grown, normally for 3 days, you image these or get add some marker of fluorescence that shows you how many cells are alive after 3 days of treatment with that drug, and that gives you an indication of whether the drug is able to stop the cells from proliferating or kill them. So the main problems that we can address with this type of system is this drug recommendation that I've kind of been talking about, and the discover relationships between the genetics or epigenetics of the cell lines and the structural elements of the drugs. But there's a big problem here in what do we mean by response to drug treatment. Okay, so there's a bunch of different ways to try to characterize whether a certain cell line responds. This is kind of the typical picture that we expect to see from a treatment with cell line. So a treatment of a cell line with some drug. So across the x-axis, these are different concentrations of the drug. For each concentration, we get one readout of the number of cells after growing it in culture for 3 days or whatever, and looking at the fluorescence indicating how many cells are present. So normally we have measures in triplicate, or hopefully in triplicate, some type of replication here. So each one of these dots represents maybe the median response at that concentration in terms of the number of cells. Then as you increase the dosage, if the drug has some effect, it will decrease the number of cells that grow at that dosage. So we kind of, from chemical studies, have seen a lot of these curves, and oftentimes they fit this type of a logistic regression. A hill slope curve goes down on the right. So in order to assess response, what studies do is look at this hill slope curve, fit it to the data that they have, and then extract one measure normally, or sometimes a couple measures more recently, on whether that cell line is really responding to that drug. Some of the ones that are often used is the IC50, which is inhibitory concentration, or EC50 is the effective concentration, and the 50 stands for the halfway point. So the IC50 is halfway between the no effect up here and the absolute zero of killing of all cells. The EC50 is halfway between no effect and the biggest effect seen. You can also look at the largest effect seen, or this theoretical possible maximal effect according to the curve, or the area under the curve, which has been more recently used as the shaded underneath this logistic curve. And as we look at each one of these different values, it would kind of change the way the shape of the curve looks. But there's a problem with using these measures in that they don't always correlate well with each other. So depending on which measure you use as an outcome, you might see different things. So this was a paper by Joe Gray and Laura and others here at OHSU, showing that the metric that you use to assess whether the drug is effective really varies across different drugs and cell lines. So this is a figure from that paper. What we're seeing across the x-axis, each of these is a different drug. And then for each drug, we have 3 different measurements: the IC50, the Emax, and log base 2 of the hill slope coefficient. And then for each one of those drugs, we have a box plot across, I think, 53 cell lines, showing the range of responses of those cell lines to that drug for that measure. And then in the top panel, we're ordering these according to decreasing IC50 values. So if I order these according to the median IC50, for instance, for this one drug, gemcitabine, it stops DNA replication. It's got a wide range of response across those different cell lines. But in the order across that, we see that there's no real correlation between what we see in the IC50 and what we see in the Emax or the hill slope coefficient. So which measure of outcome that we use is going to really change what we see here. This is some real data that from what I'm going to use for my first aim that I chunked through, just to get a sense of what these slopes actually look like, whether we can fit these logistic curves to the data and make sense of it. The first thing I want you to notice is that the ranges here are very different. So this is the raw output readings of the cellular growth after in this case 3 days, and treatment with the different doses of the drugs. So, and secondly, the replicates are kind of, in some cases all over the place, in other cases really tight together. I fit a logistic curve to this and find in this case, the IC50 value, the halfway between the max and the min. And it might be informative or it might not. In all these cases, it's not really showing us much of what we would think in terms of whether the cell line is responding to that drug treatment. Okay, it's very flat here. We still get IC50 because the mathematics is going to give you something no matter what. Here, we don't really know if it's bottomed out, right? We might have a much bigger response if we go up to higher doses. So the IC50, we could extrapolate this curve and set it where we think it might be, or we could set it to the maximum value. And different studies use different ways of fitting this curve, either with logistic curves or more complex Gompertz curves, or sometimes just linear functions, depending on what the data look like. They use different ways of throwing out data that don't look good. They use different ways of setting the values when they don't make sense with the normal curve, either setting this to the max or extrapolating the curve. So, which tends to lead to low reproduction across different studies, because they're all doing something different in order to come up with these outcomes, and then trying to match those to genomic properties of the cell line. All right, so my idea is let's get rid of all these measures, and with some fairly new mathematical techniques, we can just predict the whole curve for every cell line drug combination. And after that, we can look at these individual measures of outcome, IC50, area under the curve, whatever you want. If we can predict the whole curve, we can measure those later. So predict the whole curve, ask questions later. This is a kind of multitask learning. So instead of just trying to learn one value for each cell line drug combination, we're trying to learn, say, 9 different doses values. So what are we going to take as inputs? We'll have omic profiles of the cell lines. So this is gene expression, mutation, copy number, whatever kind of characterization of the genetics of the cell line that we can have. And for my method, we're going to have target and structural information on the drugs, so things that tell us about the shape of the drugs, what chemical structures are present, and so on. And for the second aim, something about the experimental differences between different studies, because I'm going to be combining studies. And our output is going to be the cell count at each dose. And ideally, we want to be able, these are kind of our ultimate goals of being able to handle a lot of missing data, because that's normally the case. Some of these experiments don't work for one reason or another. We want some measure of uncertainty. We'd like to be able to make predictions for cell lines that aren't in the model already, or drugs that aren't in the model already. We want to use the model to be able to identify some genetic predictors of why does this cell line respond to this drug. And if we, it would be nice to be able to learn the kind of model parameters along with the model, and some conditional dependencies between the elements of the model. So how can we do this? This is where the tensor idea comes in. This is my first picture of a kind of tensor. This is that same data that I showed some of the dose response curves for that I'll be using in AIM-1. The first frontal slice here is a matrix of values where red is higher and blue is lower, showing the count of the number of cells present after growing for any particular cell line and drug combination. So you notice there's some missing data here. And then as we move back towards the back of this cube, this is as the dosage is increasing. So red is high and blue is low, so we're getting, you can see kind of more blue in the back there, because as the curves go down towards the end. So this is the central object that we're going to be trying to fill in missing values on and predict. And it combines all the cell lines and drugs and doses into one object that can kind of leverage information of each dimension off of each other. All right, so that's a tensor. What are we going to do with this tensor? So we're going to decompose it. We're going to break it into pieces, into matrices that'll tell us individual binary relationships between one dimension of the input data and some latent factors. And by incorporating some probabilistic Bayesian methods, we should be able to do all of those goals that I laid out in the previous slide. So this is a little bit of background on previous stuff that's been done with tensors. In machine learning, these are starting to become more popular. The first decomposition method was written about in 1927, but didn't really start to get used until we have computational power to deal with these things. So starting in the 70s and more recently. But this particular study was interesting, I think, just visually, because what they're trying to do is identify a picture, identify a person based on their picture, and they separate the dimensions of that picture into the identity of the person, which is going along the kind of back axis here, and the angle from which the picture is being viewed, which is going across the front, and the light, the angle from the light, the illumination. So they're able to take a specific picture and decompose it into these 3 aspects. And then in order to identify what the person is, you just want to look at the identity portion of it. So by factoring this tensor, you can decompose these type of separate relationships within the data. Another more bioinformatics use of tensors was published in 2007 here by Orly Alter's group. She's huge in the field. And this was looking at DNA microarray data, so expression of genes over time. One of the gene expression data sets that they used was from Paul Spellman's 1998 paper, which is kind of all over the place. I mean, not over the place in a bad way, but used a lot. So it looked at expression of genes in yeast and tried to get groups of genes or eigengenes that were expressed in kind of the same pattern so that each gene could be made up of these different components of types of expression, kind of overarching themes of expression for different groups of genes. All right, so before I move into exactly my model, let's look at what other people have done for trying to predict the response of cell lines to drug treatments. This was a paper by Adam Margolin, who's here at OHSU, which was a really high throughput way of examining all the kinds of models that are out there now to see which what was most important about modeling and trying to solve this problem. So he had over 110,000 different combinations of models. So the things that he varied were the type of input data and the algorithm that was used, the type of output data, so whether we're trying to predict the IC50 or the area under the curve, and whether that output was binary or continuous. And for each of these models, they're in the category of where we're only looking at one drug at a time. There he is right now. We're only looking at one drug at a time, but all the cell lines. So we're using information from all other cell lines to try to predict the response to one particular drug, and examining what attributes of that type of model is best in predictive performance. So what they found in this paper was that, first of all, incorporating more cell line omic data, more gene expression or copy number data is more important than what algorithm you use, as long as you use a decent algorithm. Secondly, among algorithms, the elastic net or ridge regression perform the best. These are some type of regularization methods that induce some sparsity to the solution that tends to make it more generalizable. And our method, the Bayesian Tensor Factorization, with a Bayesian prior on some of the matrices in the factorization, will be somewhat similar. And also they found that the continuous area under the curve measure was the best for prediction. And they hypothesized that it's the best because it takes the full curve into account in some way, which again indicates that taking in what we're doing, predicting the full curve all at once, is a good idea. So another way to try to assess what's the best method out there for solving this problem is to fix a data set, input and output data, and then release that to the world at large and let machine learning people all compete and try to whoever can do the best job of predicting the outcomes given that data set. So that was done by the DREAM Challenge here, also organized by Laura, who's here, and Joe Gray. And the best performing algorithm was done by Mehmet Gonen and his group, which was a kernel method. And it was unique to all the methods that I mentioned in the previous study, in that instead of trying to predict the outcome for one particular drug at a time, it actually incorporated all the drugs and all the cell lines into one model, so that knowledge about the response of a cell line to one drug could help inform the prediction for a different drug. So this is a figure from their paper about this multitask, multi-kernel learning algorithm, where they combine information from all the drugs and all the cell lines through this type of matrix multiplication to this central object, in this case predicting the GI50 values across all cell lines, all drugs at the same time. They expanded this a little bit further, incorporating not just all this cell line data. So up here we have the genetics of the cell line, mutation, copy number stuff, but also incorporating outside information about the structural elements of the drugs and targets and fingerprints. So with the previous method, we just had information on the cell lines. Now we have information on the cell lines and the drugs that we're using to predict the outcome of the response between the cell lines and the drugs. And another thing that this allows at school is that we can add in a new cell line with its genetic characterizations and predict the response for a new cell line, a new drug, or even ultimately a combination of a new cell line and a new drug. So my method expands on this by kind of taking it up another dimension, adding in the response at each cell line, at least for the first aim. So this is my first aim. It's going to be a three-dimensional tensor factorization method. And the data that I'm using is the Laura Heiser, Joe Gray lab data of 70 breast cancer cell lines, 90 drug treatments, all done fairly uniformly, so I don't have to worry as much about differences in experimental procedures. So we'll compare it to current methods that I mentioned previously on these measures after predicting the whole curve. And we'll look inside the model to try to figure out if it has a good does a good job of predicting response. How does it do that? What genetic information is it using to predict response? What structural information on drugs is it using to predict that response? So this is a picture of what the models look like. The model is going to look like. So we start off with this cell line data. So we think of this as each each row here is this one cell line, and each column represents some genetic information on that cell line. So it could be the expression of a certain gene, or whether that gene's mutated in some binary marker. We take that matrix and multiply it by this projection matrix to reduce its dimension down to a smaller matrix. So in this projection matrix, here we go. So I've got one cell line, if I multiply that by a row here, I get one value in this latent matrix. So the column of the projection matrix is going to kind of pull out salient information, which genes, what expression of which genes matter, or what mutations matter, going into this latent factor. I do that across here, and I get a whole row of latent factors. So the same cell line is represented here, but instead of it being a huge matrix of all the genetic information, I have this reduced representation of genetic information, where only the important things have been pulled out, and that happens from the learning process. So the gray things here are observed. I know the cell line data. I know the central tensor of outcomes. I know the drug input data and my doses, which is just a vector. What I'm learning is the green thing. So I'm learning how to project that down to a smaller space. And these blue guys are all latent factors. So there's some representation of the gray data that's been reduced in dimensionality to go into the central tensor object. And by putting a Bayesian prior that enforces some type of sparsity on these latent factor projection matrices, not latent, but projection matrices, I can make sure that I only pull out, say, certain genes, the expression of a smaller number of genes for every cell line that will predict its outcome to the drugs. This is just a quick overview of the data. We've seen this one before. That's the central tensor. This will be some input for the cell lines. This isn't quite all the data sets that I plan on using, but it has subtype information, some gene expression, gene copy number, and gene methylation data. So this is that long matrix on the left. And this is one of the data types that I'll be inputting as drugs. These PubChem binary fingerprints, they're basically ones and zeros in this matrix. And again, I have a drug here on each row. And for each column, it's one specific structure inside that drug. So maybe it's a 6-carbon ring or a 6-carbon ring with a hydroxyl group. And it's just a 1 or a 0 as to whether that's present as part of the chemical formula of the drug. So by having this as my inputs, I can start to tease out relationships between the genomics of the cell line and the actual structural features in the drug. So just to kind of go through the math at a little bit more explicit so that you guys can be clear on exactly what the method's doing, I'm going to reduce the dimensions down to just 2D for a minute and talk about PCA, principal component analysis, which hopefully most of you have seen before. So this is just for one drug, tamoxifen. What I have here is these are basically all my dose response curves put together. And instead of seeing them go downhill, we're seeing them change color from red to blue. So the higher expression is in red, lower is in blue. So we see the kind of slopes that we expect in some of these, right? We're high here, and then we dive down to blue for the last 2 doses. So if I factor this with principal component analysis, what that really does is split this matrix into two different matrices in a specific way so that I'm capturing most of the variance in the first column of this matrix and whatever's left over in the second column and so on. If I look at just one row of this principal component, so this is my kind of latent representation of the cell line matrix across these principal components. If I look at just the top row here, what's happening? I've got a strong blue here, and then pretty much everything else is 0. That's going to get multiplied by columns in this matrix, right? So the strong blue is getting multiplied by this red across the top, and that gives me one value up here. I'm doing that for each of these values. So all the other values in this matrix don't matter much because these values are close to 0. But the strong blue is pulling out this red band across the top. And blue is a negative value, red is a positive value. So I get a negative times a positive here. This gives me a negative value or a low blue value or a white value, kind of a low expression. So what this is kind of telling me is that for this particular cell line, all that matters is this first row of the principal component matrix. And that first row is kind of evenly distributed across all doses. And it's just saying that this cell line has kind of a low expression across all doses. Not much is going on. It's just and this is really, in my mind, relates to the cell line growth rate, right? Because we're treating this with a bunch of different doses, and then no matter what dose we treat it with, there's only, there's kind of a low growth of that particular cell line at all those doses. All right. If I look at this cell line, on the other hand, though, we see that it goes from red to blue. So it's really showing a response, and it starts off with strong expression. So let's look at what is happening in its principal component decomposition. So in the first principal component, we've got this strong red. So that's pulling out a strong response from this top row across here, which means that it's, for the first 8 doses, it's got a pretty strong red response. Okay, so this is a fast-growing cell line. The second principal component also has a fairly strong red here, and that correlates to this second row. So the second row doesn't really care about the first 7 doses, but the last 2 doses have a strong response there. So that's pulling out the fact that we change from a high red to a low blue. So that's really saying something about the response here. The second principal component seems to correlate with the response, which was really exciting when I first saw it, right? We're pulling out, the most important information is just the general growth rate of the cell line, which is often normalized out in these types of studies, so people ignore it. We're capturing that information and including it in the model, and it's the first principal component. That's fine. But what we really care about more is probably the second principal component. So this is just kind of a zoom in. That's the first principal component. If I look across these different cell lines, I can see everywhere there's a red, like I kind of have a higher expression of that cell line. In general, everywhere there's a blue, there's a lower expression of that cell line. But if I zoom in on the second principal component, everywhere there's a strong red, those are cell lines that are responding to treatment. They have some, the drug is actually effective at some dose. And the third principal component is doing something cool as well. It's kind of telling us whether that drug, or whether that cell line responds to the drug early or late. So if I have a blue, it's responding at the last dose. If I have a red, it's responding a little bit earlier. So this is maybe something similar to the IC50 telling us at what dose we need in order to get that response, all being captured in the same model. And this is just 2 dimensions. So, but a problem with the 2 dimensions is that if I were to do this for a bunch of different drugs, I might not see the same principal components for each drug, right? So by combining this in the 3-dimensional tensor and doing something very similar, I make sure that I have the same type of latent representations for every drug and that they all make sense on a grander scale. All right, so how do I split apart a tensor into these types of principal components? There's 2 main methods for doing that. So this is my central tensor that I'm starting out with, the responses. The CP method was the one published in 1927. It's kind of the earliest method. The other one was just as soon afterwards. The mathematics on it is fairly old. And the idea behind this is we can take any tensor and split it into kind of simple tensors that can be thought of as just a product of 3 vectors. So if I just have 3 vectors and I multiply every possible number in every one of those vectors by every other one and put them in order, I'll get a tensor, but that tensor doesn't really hold a lot of information. It just holds as much information as 3 tensors. It's a rank 1 tensor. So I can split any tensor into a summation of rank 1 tensors. There might be a lot of these. If I cut off that summation at some point, I'll have some error left over for those that I'm not taking into account. And each one of these is kind of weighted by this central cube of how much those influence the values in the ultimate output. If I collect all the green vectors and put them into a matrix side by side, and all the red and all the blue, I get these factor matrices. And if I define a type of multiplication, I can think of this as, instead of a summation of these rank 1 tensors, it's a multiplication of these factor matrices. And those are combined through this core tensor, which in this case only has values along the diagonal. And moreover, each of these matrices has the same column dimension, because it came from just the summation of, in this case, R of these rank 1 tensors. So I'm splitting this into n by R, or in this case, I1 by R, I2 by R, R3 by R matrices. So in my experiments, this wasn't good enough to fully capture all the information that I need from my response tensor. So the second type of tensor decomposition is a little bit more flexible in that each one of these matrices can now have a different column dimension. So they're not forced to all be the same size, basically. So let's just look at some data on what that does. So if I take that tensor that I showed you earlier, and this was just to try to understand whether I can take that tensor, break it apart into pieces, put it back together, and still have all the information there that I started out with. And what size of those pieces have to be? What are the dimensions of my factor matrices that need to make sense? So if I try the CP decomposition, in this case, a 10 by 10 by 10. So I started with just 24 cell lines that I had information for 90 drugs at 10 doses. I split it into a 24 by 10 and a 90 by 10 and a 10 by 10 factor matrices with the CP decomposition, put them back together, and then looked at those dose response curves. That's what we're seeing in the top row of graphs here. So the actual observed values are blue. The predicted values are in red, or reconstructed values, really. And we can see that the red, well, they don't match well, but they did get the kind of range in general right. So it is capturing some information. This goes from, in this case, just to 3,000, while this one goes to 6,000 or so. So the red is capturing some information, but it's not enough to really see the intricacies of the curve. But if I do a Tucker decomposition instead, which in this case reduced each of the dimensions by half, well, it reduced the cell line and the drug dimension by half, so compressing my total information by a factor of 4. And then building it back together again, I can reproduce pretty well the actual shape of the response curve. Right? So this tensor factorization method, the things that really give me hope that this is going to work well is, one, PCA seems to make sense in just looking at the previous stuff I showed with factoring the doses. And two, this tensor factorization method seems to be able to hold the data and break it apart and put it back together in a reasonable way. Just to look at some of the factor matrices from that tensor decomposition, the second one, the Tucker one. This is the latent factors for the cell line and the drug, kind of hard to interpret. But if I look at the latent factors for the doses, this looks very similar to what I saw in the PCA. The first latent factor for the dose is kind of spread across the early doses and kind of tells you the general growth rate, while the second latent factor might be telling you something about whether the cell line responds to the drug or not, and the third one might be telling you whether it responds early or late. So the same type of patterns are cropping up in this method. And these factor matrices are combined through that central core tensor. These are slices through the core tensor. So these kind of tell you the weight that's put on each one of the latent factors. And similar to PCA, we get a strongest weight in the upper left-hand corner in this case, which corresponds with the general cell line growth rate. All right, so how am I going to assess whether this method works? Well, I'll compare the performance to those some of those methods that I mentioned earlier, the best performing from the DREAM-7, and Mehmet's other method of incorporating drug and cell line information at the same time. I'm going to see if incorporating dose really, on its own, improves performance much. And there's different types of prediction tasks that we can do. So we can leave out a particular cell line drug combination, all doses for that, and then try to predict it. So to do those types of predictions, I'll do a 5-fold cross-validation, so leaving out 20% of those combinations and predicting on those and then comparing to the actual observed outcomes. Or we can actually just predict for a whole cell line at every drug, how it would respond to every drug. And this is really what we'd want for a type of personalized or precision medicine. We want to know for a new patient, how are they going to respond for all these different drugs. So for that, I'll leave out one cell line and make predictions across all drugs, or one drug and make predictions across all cell lines and compare. All right, so moving on towards my second aim, where I'm going to try to combine multiple studies in a slightly larger tensor to make sense of this type of data done from multiple studies at the same time. So these two papers, which came out in the last 2 years from John Quackenbush's group, are really kind of calling attention to the fact that there's been several of these large cell line drug panel screening efforts, and they haven't necessarily reproduced the same results. So this is one figure from their second paper, just kind of showing the differences between the studies and why. So this is comparing two studies, the CCLE, or Cancer Cell Line Encyclopedia, and the CGP, or Cancer Genome Project. And they have kind of moderate concordance in how they see their gene expression, but low concordance in what they see as responses to different drugs, like which cell lines will respond to which drugs, and low concordance in between the relationships that they find between the genetics and the drug response. And some of the reasons are these red boxes, what's different between the two studies. So there's a lot of differences in the way that the studies performed, how they grow the cells and plate them. And I think, well, one of the things that my method will address fully is this differences in how they model the dose-response curve. Right? They use different methods for fitting the curve and different measures of response between the two studies. So this is my second aim, to combine, in this case, 4 studies in now a 4-dimensional tensor that's going to capture all the information of these studies and use them to try to understand why there's differences between these studies for the same type of prediction outcome. This is an overview of the data for the 4 studies. So the first one will be the data from AIM-1. These next 2 big studies are the ones that I mentioned in the previous paper. They have hundreds of cell lines, hundreds of drugs. So it's a large amount of data to deal with, but these tensor methods should be able to handle this fairly naturally. And the CTD2, which I forget what the acronym stands for, Cancer Target Discovery and Development, is another large study with a lot of some overlap with cell lines of drugs of the previous one. So here's some of the overlap. I didn't include the data from the first study, which I don't know if I was clear about. The data on the first study is just breast cancer cell lines. These studies are across multiple cancer types. So the overlap, there's some overlap between, for instance, the CCLE and the CGP or Sanger data. And the method, the tensor model, should be able to leverage this overlap for predictions and to understand what elements of the experimental differences are really accounting for the differences found between these studies. Here's what the model looks like for AIM-2. It's very similar to the previous model for AIM-1. The differences are I've added in an extra dimension here that incorporates, that summarizes the differences in the experimental conditions between the studies. So there'll be a row here for each of the 4 studies and columns kind of indicating what growth media they use and different experimental differences between the studies. So that those experimental differences can now be linked to the drug response data as well. Also, the central tensor is now this 4-dimensional hypercube. And the last aim, is that clock right? Oh, yeah. Okay. So I'm going a little bit fast, I guess. So the last aim here is to try to get towards some of the actual biological evidence supporting what we find in these studies. So I'm going to look at this RNAi screening data from something called the Achilles project. And I'll look at published cell line pathways in a database to try to look at connections between the genomics and the drug features. So about this Project Achilles, this is a totally separate type of data which should be able to, which we can use since it's a completely separate platform for some type of validation. So what they do here is they infect cells and a good amount of the overlapping cell lines from what we saw in the previous aim. Infect cells with short hairpin RNAs, which are processed into small interfering RNAs, each of which targets one specific gene. So the kind of orange plasmid here would target one gene and stop its function. The green one would target a different gene, stop its function, and so on. And then they grow the cells. And then they count basically how many cells are left after growing for a certain amount of time. So if the orange gene is important, when we knock it down, we're going to have less cells grown over time. That's why it's called Project Achilles. They're really trying to determine which genes are important for that cell to grow. So we're kind of looking at something similar in our study where we can look at literature to see what genes a particular drug targets, and then we can see whether mutations in that gene or expression of that gene in the cell line is correlated with whether that cell line responds to that drug. So for a given drug here, that gets projected through to these latent factors that incorporate some of the information on the structural features of the drug, which is somewhere in this core tensor. That latent factor, we can examine which latent factors about the cell lines are most highly weighted against the information from that drug, and trace it back through the projection matrix. And these ideas of kind of eigengenes, of groups of genes that are regulated together or have the same effect on specific structural features, and compare that in using permutation analysis to see whether these are represented, whatever genes are pulled out by this projection matrix are more often represented than would be expected by chance in targeting that particular drug. So the database I'm going to use for this is the CPDP, which is a compendium of a bunch of different databases. So that's the consensus pathway database that pulls together 32 different databases with over 4,000 pathways, which I'll be able to examine to really look for evidence of whether the relationships I find in my model have been found in literature previously, and whether relationships found in literature previously are overrepresented in my model in terms of these permutation analysis. This is the timeline, so I hope to get this all done in around 2 years. I want to publish at least 2, maybe 3 papers off of it. The first aim in this first tensor method is more of a technical paper. And then the second one, really trying to make sense of those disparate data sets, pulling them together, I think will be a bigger, more bioinformatics paper. That might be kind of boring. And just overall an impact, what is this new method going to do? Okay. So for aim 1, we've got this whole new method. We'll be able to predict responses for new cell lines and new drugs, and also tease apart some of these relationships between cell lines and drugs. Aim 2, we're going to combine these data sets that until now it's been kind of worrisome that they haven't been finding the same things. So now we're in a unified framework that we can make sense of them all. And aim 3, we're really going to look towards mechanism. Is what we find with this method supported by literature and RNAi information? And all the code that I write is going to be written in R and shared freely with the community. And if you just take a second and think about it, as my last slide, I just want to think about some extensions and how broad this kind of framework is and how it could be used for a bunch of other things. So kind of on the first level, we could have all kinds of different input data on the cell lines or the drugs. We could include a lot more descriptive things about the epigenetics of the cell line. We could even have properties of imaging data from what the cells look like grown in different media and so on. We could incorporate that RNAi screening data as well as input into the cell lines for our predictions. And any of these might improve the prediction a lot. Also, we can think about what other dimensions could we have on this tensor? Okay, maybe the cell line is not responding to the drug just because of its genetics or because of the experimental conditions that I put in there. Maybe there's something about the microenvironment in which it's grown, and we can add an extra dimension detailing the surrounding of the cells if it's grown in a different type of culture or a different extracellular matrix or different signaling factors around it. And there's a big project going on starting here at OHSU through the LINKS grant for this type of data, this microenvironment, microarray, where there will be growing cells in different microenvironments, and seeing how those microenvironments influence the growth of the cells. So that was one extension that I had in mind from the beginning of the study, and hope to work on that data as well once I have this framework set up. And I know there's some clinicians out there. You can really see this being used directly for patients. If we had, instead of outcomes, these measures from cell lines, we have some direct measure of clinical outcome. We can have as input dimensions, obviously genomics, but also possibly the environment of the person, maybe even something about their microbiome, or the treatment schedule, or dosage of the drugs. There's a huge number of different factors that we could try to incorporate into one of these tensors in order to make predictions on a broad number of different categories, different types of problems here. So that is all I have. I'd like to thank my committee, my department, DMICE, NLM Fellows for letting me practice my talk, Coborne Lab for also letting me practice my talk, Joe and Laura for meeting with me and giving me advice on the project in general, NLM for funding, and of course my friends and family out there. And ask for any questions. Hope I didn't go too fast.
Just a reminder, if you ask questions, because it's being recorded, especially for people watching it live, just press the button and hold it down while you're talking.
Is all totally clear? Yes, James.
Wow, great talk. Thank you. Are clinical decisions being made today on some of these studies that have not been replicated?
I don't know that. I don't think so, not yet. Some of these studies have replicated things that have been found. So basically, they're normally used as screening studies, where you'd want to, if you find a relationship between some genetic feature and some drug response, you'd want to examine that further with a clinical trial. Well, a few steps before you get to the clinical trial. So these cell lines are fairly early in the pipeline in terms of drug development.
But it does have an economic cost, because, and Bayer and others have done internal studies in which they've seen that over a quarter or more of the portfolio can't be reproduced. And those are all things that continued on in the pipeline. So that has a really immediate aspect in terms of either choices and other things that could have come in, or the cost, or both, right? So very nice talk. I have a question for you. I was thinking about it, and I should have brought it up earlier, but we have a lot of information more and more now about the actual specificity of, or promiscuity of the drugs. And I'm wondering if you're actually explicitly incorporating that. I know I brought up before the idea of the relationship or the correlation among the drugs, because many of them can be related. But I was just wondering if you're explicitly looking at that, because that could be interesting, especially when you're thinking about error, right? Where you have drugs that are either highly specific, or you have drugs that potentially have a lot of off-targeting or other aspects where you would kind of see kind of cross-signal or noise in some of the other cell lines where you might not expect a response, and you're seeing a response.
Right. So you're saying this will increase generally the noise in the response to that given drug, or maybe that drug will have higher specificity in a certain genetic background than in others. I think that those type of relationships, so with this tensor method, we should be able to, we will be able to have some measure of the variance across the measurements, right? So depending on how I train it, and I have a couple different methods that I've been thinking of. We'll have some measure of not just what we expect for the outcome at each dose, but what's the variance that we expect for the outcome at each dose. So we can connect that variance to drug properties as well. And for specific drugs, we can push that particular column through the tensor and try to see whether the variance correlates with certain features of the drugs, or for specific drugs, whether there's higher variance in the outcomes.
But I think, I don't want to speak for Shannon, but it's, I think she was asking whether this can be used as a side information, because you know that, and can you use that to restrain or constrain your model for better prediction? Or this is my follow-up at least, so this is what I understood.
So as an input to the drug kind of matrix, some information about the specificity of the drug as a predictor. Yeah, I think that's a really good idea. But I'll have to think about where I would find that.
We can talk
Yeah.
offline, but I think it actually could be really informative, especially because some of that data is good and some of it's actually prediction itself. And so it could be really interesting to incorporate it and then be able to also assess, especially some of the ones that are more computational rather than experimental.
Does this work? Have you ever thought about adapting it to incorporate sort of a continuous update of time of your variables? So, for example, usually when we think about patients in an EHR database, some of their variables are continually changing and being updated, and we might one day capture a continuous stream of biometric data and then say apply your method, and it might be adaptable to that kind of data in the future.
Yeah, I think that's something that I probably should have put on the extensions, right? That's a really good point. So a lot of the tensor methods, well, a lot of uses for tensor methods have incorporated some element of time. So one study was uses tensor factorization to predict movie ratings, but they also incorporated a time element. So maybe this movie's more popular at this time, and then later not so much. So yeah, definitely, I think moving towards the clinic, that makes a lot of sense, and especially with this kind of multitask prediction, we can predict at each time point or use the previous time points to predict the next one. Similar as we might use the previous doses to predict the next dose or output here.
It'll be your postdoc.
Yeah. I mean, there's so many extensions for this. I'm really excited about the idea in general.
Or yours.
Yes, Cesar.
So first of all, great talk, and I think you set the bar really, 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 really high out. So I have two questions. One is, if you go back a few slides where you're talking about your tensor and the dimensions of the various things the matrices that are going in it, keep going. Maybe 5 slides back. Yeah, sorry about that.
Okay.
That's good enough. One more forward is good enough. So how do you make sure that the dimensions of the matrix that are going into your tensor are appropriate for that tensor factorization?
Right, so that's kind of what I was getting at here, right? So I tried some different sizes decompositions. So this was reducing the data basically by a factor of 4, and I was able to get good reproduction of the outcome values. So that's basically what I'm going to use as the indication. Without being linked to the genomics and the drug features, just can I reproduce the data that's in the tensor? Okay. Then I'm really holding enough of the information in these factor matrices that's a decent size. So I plan on, when I write the script, trying it with different sizes on Exacloud in parallel, and finding which ones seem to have the lowest error when I reproduce the central tensor.
So you're going to set a curve where you get it to that minimum or something, that you optimize it to that point?
Yeah, we're gonna optimize it exactly. So, yeah, we'll have for different values of this decomposition, different dimensions. We'll look at the error function. I mean, another way to do it that's more complicated is to try to do it with Bayesian methods, where you have an objective function that actually takes into account that error and something about those dimensions as well, and it allows it to change the dimensions while you're training, which is another possibility. I've seen some papers that do things similar to that. But, so that might be a further down the road development of the method. For now, I think I'm just going to try a bunch and see which one kind of is able to reproduce the central tensor, so I'm sure that I'm capturing the full amount of information.
Thank you. And I guess my second question is, how do you deal with cell line generation? And what I mean by that is, as cell lines move through time, and which generation they're taking sampling from, they might differ. And so cell line BT474 might have a different drug response when it was first thought out in the first 20 days versus a 40-day in culture. How do you deal with that?
So that is a problem, and that I think will. So for the cell lines that are existing in 2 different studies, I think that's where that'll be the most problem. In my second aim, where I'm combining different studies, and they have different versions of the same cell lines that might have been growing separately for possibly decades at some time. Like people have seen, Andrew Adey did the genome on HeLa, right? There's a lot of differences in some of these HeLa cell lines that have been growing forever. So that's some possible difference between these. So one way to incorporate it in the second model is I'm going to have characterizations of the genetics from each of these different studies that can go into the cell line matrix as a prediction. Okay, so I won't just have like a block of expression data for cell line A. I'll have block of expression data for cell line A from study 1 and block of expression data from cell line A from study 2. Right? So, and with the sparsity prior on the projection matrix, I will hopefully be choosing, it will choose one or the other of those expression data to use as its prediction passing through the rest of the model. The characterization, that's a good idea, and I should look out for it, for differences in whether it's going to choose one or the other of the expression data from the different studies. And we can compare those expression data. Like when the figure I put up from the paper comparing studies showed moderate concordance in the genomics between the different studies. So yeah, it's something to keep an eye on, I guess. And there's also been some recent papers about cell line contamination. Some of these studies have done a good job of making sure they're not contaminated and have gone through a lot of steps to see that each cell line is different, and some of them less so.
You could, though, directly incorporate it as metadata into AIM2 if you have that information. The problem is in all studies you won't, but if you actually know passage or if you know purity or if you have any of that, that could easily be added as well.
Yeah, so, right, in the experimental differences matrix, yeah.
Any other questions? All right, just join me in thanking him one more time. Thank you.
I wanted to ask.
So the committee has to meet
Do you
now?
know who would be a good reference for learning about doing process maps and for using tools that assist in building process maps? Because I talked with Paul, he recommended Carmeet, and I think that that's what I'll have to do in order.
Carmeet, really?
Carmeet McMullen.
Yeah, I know. Process maps?
Apparently she does process maps, and he recommended her. But I think that I will have to build process maps, and what I was planning to do was to get video of, clinical teams doing simulated code, hospital codes, and I want to build the process maps and analyze this data orthogonally, like same team, different sims, and different sims, same team, so I can analyze changes to the structure and then changes to the state and dynamics of the system.